Okay. So it's weird that I have to introduce myself, but I just want to say something which is important for the course because I didn't say before. So I am professor at the Department of Pathophysiology, the Clinical Hospital, the School of Medicine, Universidad de la República. And I have a shared position between the Universidad de la República and the Instituto Pasteur as the head of the Advanced Bioimaging Unit. And basically, all my motivation is due to the instrumentation, some methods related to spectroscopy on images. So I will talk now basically about some details of instrumentation related to fluorescence. So, well, as you know, I mean, the opportunity that brings the fluorescent microscopy is unique because we can go from a few millimeters to tens of nanometers, covering a whole range of different techniques, from diffractive limit microscopy to super resolve microscopy. And fluorescence is really a powerful tool in order to find the particular object that we want to find within our system of study, like well, viruses, bacteria, cell, animals, etc. So we know that basically we have to handle with the problem of the resolution, which is due to basically the optics we use, as well as the wavelength. And at the end, no matter what, we will have a resolution which is limited in the axial and the lateral, depending on these two variables. Well, the numerical aperture of the objective that we use, as well as the wavelength. And if we use probably a visible range wavelength, our resolution, no matter what, will be on the range of 200, 300 nanometer in lateral and 500 to a micron in the axial. So, but the good things about fluorescence is that basically we can use different approaches like wide field fluorescence, in which we focus the light within within the optics. Unfortunately, these have a lot of out of focus fluorescence, and this is how it looked like, for example, when we do wide field fluorescence. In comparison, confocal microscope will reject the out of focus, and we will see how in a minute. And this is important because we can cover different region in the cell with zeta stack resolution. This is also done by another technique, which is called a spinning disk, which is kind of a similar approach of the laser scanning confocal microscope, but with lower resolution. But as usual, the limitation in one side are advantages in another. For example, in this case, you can go really fast scanning multi-point and basically have a resolution of the rate of the camera. The other possibility is, for example, TIRF, which is an effect that you can excite fluorophore near to the surface, and these are very useful when you are studying things in the membrane. So the most novel techniques related to super resolution that can be split in three. I will not speak much in detail because we will have a very nice lecture, couple of lectures on the Thursday by Luca Lanzano and Fernando Stefani, are related to, well, the true super resolution, functional super resolution, or stochastics. Basically doing different kind of approximation, we can break the diffraction limit, and today we can go up to nanometers, as we will see in these lectures. So, but what we need for doing a fluorescent microscopy? Well, of course, we need a source of light. So in our case, for confocal raster imaging, we will use lasers. We will need to move the beam, so we will need to use a scanner or some other strategies like piezo stage. I will address all of the options, and then we will need to detect the light. So for this, we will have two main groups, I will call, basically one related to point and the other related to many points in parallel. So the PMTs, APDs, or the cameras. And then I will cover some other stuff about little things about optics and electronics that we need to do this kind of approximation. So when we compare wide field microscopy versus confocal microscopy, basically for the wide field, we use a source of light that can be an halogen or a mercury arc lamp, and we shine the light, and we have here a dichroic mirror that can reflect the light, and then a filter who select the wavelength that we want to use to excite our sample. And in the way back, this dichroic mirror enable the pass of the fluorescent light, which is usually larger, longer wavelength than the excitation. Then we will have another set of filters that we collect the band that we want to collect, and then we will have different configuration, our IPs or cameras, different kind of camera with different path. You may notice that here we will collect all the light coming from, up and down of the focal point that we are interested in. So to avoid this, the confocal microscopy use an object in the optical path, which is called the pinhole. And the pinhole rejects any point which is out of the focus what we wanted to see. This is really a great advantage, and basically changing the pinhole, we can change the resolution, but of course, if we close too much the pinhole, we will restrict a lot of light, and we will be unable to see anything from the sample. Considering that we have the pinhole, of course, we will be acquiring a point, which is called the point spread function. So we need to basically move our beam, and for instance, we need basically a scanner. So basically, during the imaging technique, we use a laser scanning microscope, and this can be used in regular confocal microscope, super resolution microscope, as well as multiphoton microscope. And here I place some examples. Basically, the scanner are a set of two mirrors who are moving orthogonally to each other, and they have different configuration depending on the kind of the scanner, and I will cover in a minute. So here are different brands from my confocal microscope. For example, here is an Olympus. This is a FloorView 1000. These are the one that we have here is the size 880, and there are many others. And this is an ALVA microscope for STED that they have at the Laboratory for Fluorescence Dynamics. This is a nice instrument for do super resolution. So for the scanner speaking, well, we have basically two sets that I will discuss. One is related to scanning a single beam or scanning a multi-beam. So for the scanning a single beam, as I said before, we have two mirrors which are moving independently. One will move the beam horizontal and the other will move the beam vertical. And this will allow us to basically do the Raster scanner across the image. So the good or the advantage of this method is related to the good quality of the images and the high resolution that we can get. And one of the disadvantages is the slow frame rate that we can get, which is on the order of 3 frames per second. This depends on the kind of scanner, and I will discuss about raster scanner versus resonance scanner, which enable to go a faster rate. The other possibility is to use a multi-beam, and for this, it was designed the spinning disc confocal microscope based on the Nippon's disc, which has basically a micro lens and multiple pinholes, and we can collect light coming from different places in the sample at the same time, and then acquire all of them in the camera. This is going very, very fast because we can read many of these pinholes during the time that we are acquiring 1 frame rate. And the thing is, one of the advantages that we have are the video rate that we have. And the bad thing is the low resolution that we have during the acquisition, because we cannot have exactly the same size of the pinhole. And this is related with that the pinhole is fixed in the size. The two kind of configuration during the raster scanning confocal microscope are, for example, the scan mode, in which we shine our light as usual with the dichroic mirror to select excitation through the emission, and then we have the couple of the mirror from the scanner, our scanning optics, the light goes to the sample, get excited, our fluorescent came back, and pass through all the scanner back to the detectors. In that case, it's called the scan because the beam after passing back from the scanner is always most of the time set all the confocal microscope that we have in our laboratories. One of the disadvantages of this is as much optical pass we have, of course, we will lose light in each of the surfaces, something that we need basically to handle with scan configuration. And this is possible, for example, in the multiple photon excitation because intrinsic confocal. For instance, what we can do is use the scanner. We still pass through our scan optics, and then we can split the light from the excitation and emission before coming back to the scanner, and our beam will be no longer stable because it's not canceled by the two mirrors. So basically the beam here is moving, and what we need to do is use optics right in front of the detector to focus as much as we can the light on the active area of the detector. This is very good when you want to save surfaces because we can put our detector right after the light coming out from the sample. And this is, I know, a couple of instruments with this configuration that work pretty good. Well, we say that we need to use light, and for confocal microscope, we use different kind of lasers, such as the helium neon, argon laser, diode lasers, and more recently, diode pump solid state laser, which are very stable and very cheap in comparison with the previous one. But all of these lasers are continuous wave, which means that we have more or less an intensity, a constant intensity over the time. And these are not able to use for measuring fluorescent lifetime because we need pulse laser or modulated laser that we can do for different approaches, like using acousto-optic modulator, some other alternative, pocket cell, etc. But I will not go in detail on this characteristic. For this film application, we can use supercontinuous lasers, which are, for example, the white laser that they have at the LFT, the Fayanium, and also this other from YSL. They are very stable. They cover a long range from UV to couple of microns. They are really powerful, and they are intrinsical pulse at different frequency. For example, this one is a 20 megahertz frequency. Moreover, we can have also multiphoton excitation, and in the market there are different options. The good thing about multiphoton excitation is that we move our excitation to the near infrared, and this is very useful, as I will explain in the next slide. And they are really powerful. And basically, we have several brands. The new ones from Coherent are the Chameleon Vision 1 and 2. Spectra Physics have several different options. The one that I pick here is the MiTi Deep Sea with the Deep Sea model, which is a model to correct dispersion of the pulse passing through the optics. And in this model, EHP is the one with a short pulse, which is very useful for increase the excitation. This is the one that we got here to build the diver, and I will address this at the end of the talk. Then Thorlab came up with this Tiberius, which is the most cheap of the others. It covered a short range from 710 to 980, but for most of the application, it's a very nice instrument. And there are different companies. This is from Kalmar. The Carmel 780 is the one that we got for the other microscope that we are building. Is a 2-photon inverted microscope, and this is a femtosecond fiber-based laser, which is very nice. It has only one single wavelength, but is really 1/3, 1/4 of the value of the large two-photon excitation, two-photon laser, sorry. Then there are some others, two-photon laser based in different crystals that can cover a longer range. For example, these two can go from 690 up to 1250, 1300 or more or less in the two. I have the chance to use this inside in the LFD. It's a wonderful, very stable laser with a lot of power, and it's really fast switching the wavelength. It's a nice instrument. What I want to address here is what is special in two-photon excitation? Well, if you go the easy thing, which is go to Wikipedia and ask what is two-photon excitation, the definition says that two-photon excitation coefficient is based on the idea that the absorption of two photons of the half of the energy need for one photon excitation can also excite a fluorophore in a quantum event. The process, the two-photon excitation process, depends on the quadratic function of the light intensity. And for instance, we will call these nonlinear processes. And this struggled my thought about how this happened over the years. And until I put time, I never understood why this happened. And well, as Dave explained before, we have excitation in the Shklovsky-Perrin diagram, and then we have some electronic rearrangement before the emission. Well, if we talk on time, the excitation is usually on the femtosecond time scale. Then we have relaxation on the picosecond time scale, and the emission takes on the order of nanosecond time scale. When you thought about this, you notice that if you don't have two photons within this period of time, basically to hit the first intermediate state and the second before the first one will release the energy, this will never happen. And this basically give me the final hit to finally understand this. Well, if you really want to put two-photon excitation, you need to do something, because if you wait for the probability to have two photons, this will take forever. So basically, Maria Goppenmeyer described all the mathematics about this, and basically what is important here is, well, on the contrary of the continuous lasers, two-photon lasers are pulse, as I said before, and the width of the pulse is usually on the 100th of femtosecond. And this is basically very important because we need a lot of power concentrated in a pulse, concentrating in terms of time, and then focus with our optics to the point. And if we achieve all of this and we apply the power enough, we will get two-photon excitation on the spot and none above below of this point. So this means that two-photon excitation is intrinsically confocal. And what is special about two-photon excitation? Well, the first thing is basically we have an increased penetration because in the range that we use for two-photon excitation, there are is the minimum of the absorber that we have in tissue and in our samples, our classical sample, biological sample, but also is also a window where the scattering coefficient is low, relatively slow. So as increasing the wavelength, the scattering is inverse to the power of 4 of the wavelength, and this is important to basically penetrate deep in the tissue. The other important thing is the avoidance of autofocus, and here are basically the excitation with a regular wide field, and this is the two-photon, and you may see that there is no excitation up and down of the point, which basically is important not only for the confocality, but as well to avoid photobleaching above and below or the point where we want to image. So if we come back to the scanner, well, as I said before, we have a Raster Scanner, which are fast and simple. Well, we have two ways to scan the beam, sorry. We can scan the beam with the scanner, where the sample is stationary. And there are different kinds of scanner. The galvanometric is the most common. The resonant are not that common, are a little bit more expensive, but they are faster. The difference between these both is that in the resonant, we have the mirror moving back and forth with a certain frequency and not stopping, and this allows you to go faster. As usual, one and the other have different advantages and disadvantages. Galvanometric are very precise, resonant are not, sometimes are not, the new ones are much better. Then we have also Polygon Scanner, which is like this one, and we have also the use of acousto-optic deflector that can move the beam across the sample. So this is the one that we have here in the two instruments that we are now building, the diver microscope that Enrico will talk tomorrow, and a regular inverted epifluorescent detection 2-photon microscope. This is very nice scanner from Cambridge Technology, which has 10mm mirror, which is a very nice room to accommodate your beam, already span, so you don't need to deal with the scanning optic to fill up the back aperture of the objective, which is very useful. And it has a 40 degrees excursion angle, and as I said before, it's really fast and it's very linear. In the market, there are different sizes of the scanner. The most common are the 7-8mm, and in the diver in California, for example, they have the 50mm, which are very, very large to accommodate larger beams. The other possibility is to scan moving the sample, and this can be done using piezo stages, in which we can have really, really good precision in the movement, nanometer resolution, and we can move across the sample X, Y, and Z. The only constraint is, of course, it will take more time. So let's move on to the detectors. And considering the detector, I will speak first about the point detectors. And we can split basically between two main groups, the photomultiplier tubes and the photoavalanche photodiode detectors. The first one can be found with different coverage, like bi-alkali, multi-alkali are the old ones and the more common in all the confocal microscope. And the new one are the gallium arsenide photodiode, which you may see are better in the resolution. One of the important things is usually all these detectors have really large areas which make much more easy to basically focus the light in the detection zone. On average, all these PMTs are on the 30% of the quantum efficiency, and this is a comparison between the different surfaces. They have different response in the spectra as well as in the sensitivity. And you can see here that the gallium arsenide, which is the red one, is the higher, and you can go up to 50, 70, even a little bit more quantum efficiency, which are very useful for most of the photon counting detection that we do using film. Then we have the Avalanche Photodiode that can cover also a long range as the PMTs, depending on the material that we place in the active detector. And the most important thing is the quantum efficiency of those are very, very high, much better than the PMTs. And you can see this in the response that we have at, for example, 600 or 650. The problem is, of course, you have, depending on the surfaces, different ranges. And if you compare, for example, this APD with the gallium arsenide photodiode, which is this one, you may see that the range is much larger for the gallium arsenide photodiode. So how photomultiplier applies work? Well, we have a cathode and an anode, and then we have a series of diodes, and basically this is under the vacuum, and we basically, when a photon hit the surfaces, it will produce photoelectrons, and after the photoelectron will hit the successive diode, it will amplify the release of new photo electrons. Usually the multiplication is on the range of 4 to 5, but can go up to 18. And after all the step, which usually are 8, you can amplify really quite a lot the signal coming from a single photon. These are most of the configuration that we have, and depending on the kind of instrumentation that we can do, we can have this side-on photomultiplier, head-on. There are different variants. They are useful depending on the kind of spectroscopy that we can do. While they are extremely sensitive, they can be really fast on the gigahertz rate. They have a fast response, which means the time to take from the photon, the active surface at the end to the anode. And this is the common thing, as I said before. The other thing is the Avalanche Photodiode. And in that case, the physics is a little different. We have a semiconductor based on silicon. They can dope with different molecules and metals, and this can give you different response in spectra. But basically, we have a positively doped region and a negative doped region, and then in the middle we have a neutral charge depletion region in which we can apply this reverse bias, which is the potential, in order to increase the number of secondary photoelectron produced after the first one hit to the surface. And I found very interesting this animation in the web page that you can play around with the number of photons hitting the surface as well as the reverse bias, and you can see that if I increase from 1 to 10 the reverse bias, this increases the number of photons that you can, the amplification that you can find. Well, as much as you increase this reverse bias volt, of course you start to increase the dark current, and this is well, the bad side of this. I didn't say, but PMTs are very good in the signal to noise ratio. And more recently, there are some new kind of hybrid detector in which they combine the two technology, first amplification and then the APDs, and this increased quite a lot the sensitivity, taking the best of each of the technique, and this increased a lot the quantum efficiency, and you can read really fast. These are ideal for lifetime measurement as well for FCS because it doesn't have after pulsing, which is a problem when you want to do correlation experiment. So then we move to the parallel detector, and I split in two areas, two sets of detector. Basically, the classical cameras that we will see in a minute, the CCDs, the intensified CCDs, the electron multiplied CCDs, and the scientific CMOS. But these are the most common used for basically all the wide field, spinning disks, total internal reflection, light sheet microscopy, and many more. The main difference with all of them, I split basically this intensifier because you can use this for film measurement. The other set of cameras I call Nano Camera because these are actually PMTs or single photon avalanche photodiode arrays, which can be run as a nano camera, and you will see in some of the lecture that we can use, for example, the ID detector to see correlation within the different actuators, and this will be a talk by Lorenzo Scipioni tomorrow afternoon, I think, yes. And then he will talk also about the new microscope he is building, about 32 channel for spectral, which can be also used as a nano camera. And there are a bunch of people working in the field using this SPD array, for example, in the Vici Domiti lab at the Italian Institute of Technology. They're using this for fluorescent lifetime imaging on a 25 array sensor, which is very nice. While speaking about the difference between CCD and CMOS, well, these two, the main difference is how the data is read, and also how is the sensitivity and how is the size of the chip? Well, in the CCD, each of the actuator, which are usually diodes, can be excited, and basically we have a shutter which is total. So we will not be able to acquire a new image until we read all the data. And the data is coming out one by one to the amplifier and the charge to voltage conversion. And until we get out the last pixel, we will not be able to take a new measurement. And this is the main difference between the CCD and the CMOS technology, in which we have here a rolling shutter. Each of the actuators are independently. All of them have their own amplification and conversion. And basically, you read all of them line by line, one after another. And this makes a huge difference in the speed that you can read the two. And this is see here in the table that you can go, for example, in a regular CCD to 17 frames per second. And with any of the scientific CMOS, you can go up to 100 frames per second without any problem. Well, in terms of noise, these two are kind of similar. Usually CMOS are more noisier, and the noise in this case is all the noise is different because all the pixels are different. In here, basically the noise is very stable along the all pixels that we have in the camera. But nowadays, there is in the market new cameras, for example, this one from Photometrics, which is back illuminated, and you can go up to 95% of quantum efficiency, which is really a good competitor for the EMCCD's camera for single molecule. And you can see here the difference between the two resolution and the signal to noise in these two cameras. So in the electron multiplying, they have electronic that amplify basically the output of the signal, and this enlarge basically the signal to noise, which can reduce the noise basically to less than 1 electron in this. And you can see the difference between the three, the CCD, the CMOS, and the EMCCD. The cons of this is, well, usually the pixels here are larger, and you still have the time that you can read, because no matter what, you have to wait until the last pixel can read to get the new measurement. So then we have some other parts which are very important for the microscope, and these are electronics. For handle with the data, for example, for lifetime measurement, we use Flimbox, and these two technologies, Flimbox this is based on Spartan 3 and Spartan 6. These are ISS Flimbox that can handle with the lifetime measurement. And this is the new technology based on the Kintex, which is Spartan 6 car. And these are very nice car that you can make parallel measurement. And Lorenzo Scipione will talk about this in his presentation on Wednesday about the spectral film instrument that he had built. And also we need basically some optic to split excitation and emission, and I will speak in a minute about this. To read the scanner, to move the scanner, we also need cards. And usually, well, most of the microscopes come with this 3-axis card in which we can handle with the position of the scanner and move the scanner to Raster Scan. We use here the IoTech card, which is a very cheap solution, and you can do it by your own. And this is the way that we solve how to handle with the drivers of the scanner, connect with the microscope and make the microscope work. Speaking about the signal, so the signal coming from the detectors, as I said before, we can use basically steady state measurement, and the IoTech card is also useful in that sense. There are different options, like for example, FCS card from ISS. There are so many others in the market. And as I said before, film card are used for frequency domain measurement, and Becker and Hickel also have some other for time domain, which are based on the time to correlate the single photon counting procedure. And the new ones are these 32-channel FPGA developed by Film Lab in Italy based on the work of Alessandro Rosetta. But if we are talking about analog detector, we need to condition the signal in order to have a photon counting regime. And for this, we need amplify the signal, and we need to include quantum fraction discriminator, which are need to transfer the data from analog to digital and then get into our car. And this is basically the sequence of the electronical step that we need to use for the microscope. So finally, I will just make a mention about the optical filters that we use. Basically, we usually use dichrome mirror, which enable to basically split signal depending of the wavelength. They can be short pass, long pass, or band pass. And then we use usually band pass filter that can be based on two technologies: interference filters that are deposit of different layers of material that can interfere and enable or not the pass of the light, or colored glass filter, which absorb and reduce the transmission for certain wavelength. So finally, I would like to comment the instrumentation that we have here at Uruguay, and in particular to the Institut Pasteur, and as well as the Hospital de Clínicas, where I am located. Well, in Uruguay, we have several confocal microscopes. For example, in the School of Medicine, they have the SP5. This is a spectral microscope with several lines and excitation. It has a resonance scanner and a galvo scanner. This enabled to work with CO2, temperature, and many other adds that they include over the time. They also have fluorescent capabilities. The Instituto de Investigaciones Biológicas Clemente Estable, they do have two confocal microscopes, and all upright, still working confocal microscope from Olympus, and also the new one, LSM 800 from SAI with the ID detection, which is a very nice instrument to do sort of super resolution that we can reduce to the half of the regular confocal microscope. And also it has the cabin for CO2 and temperature, and they do have also an epifluorescent microscope. At the School of Engineering, we will have on Thursday, basically a participant from the School of Engineering. When they are building new microscope, basically one which is called an epifluorescent microscope with a multifocus epifluorescent based on electrical lens. So the sample is not moving, and they are moving basically the focus where are looking with the electrical lens, and we will see how this works in some other talks. And they are now working on SPIM with adaptive optics for work with cells in a large view. Here at the Institut Pasteur, we have two confocal microscopes, one 800 and one 880, which is a pseudo-spectral detection, because we can collect the spectral detection step by step along the range that we set up. And then we also have an epifluorescent detector for simple measurements. So the two projects that I am working on now are, one is, as I said before, an inverted microscope based on this Carmel 780 Faber-based 2-photon laser, and then it will be built on top of this Nikon TI-S microscope. We got basically motorized stage, the scanner that I speak before, detector that I talked before, and the ISS fast film in order to do film measurement, also phosphorescent lifetime measurement, FCS, 3D orbital tracking, and most of the technique that we will cover during the course. Then we are working on in another 2-photon microscope. This is the famous diver microscope that Enrico will introduce on Wednesday. And this is a unique microscope for deep tissue imaging, harmonic generation, and autofluorescence. It's based on a large PMT in the trans detection. And we will see how this works. It's a very nice. It has a really large scanning optic that enables to have large field of view, and these are very useful for in vivo or for large tissue samples. And we will use this beautiful two-photon laser, which is the MITI, as I said before, with the deep sea model and the EHP model with a shorter pulse, which enable to increase up to 3-4 times the excitation efficiency in depth without increasing the power laser. And this is also important when you work with the in vivo samples. We will use the same scanner as the other, and we will be using this new 4-channel film box based on the development of Alessandro Zeta from Film Labs in Italy, which are really nice card that you can go up to gigahertz of frequencies.